This video will look at bacteria growth and look at how bacteria growth is influenced. Okay, so bacteria can be found almost everywhere. It lives on us, lives around us, it lives pretty much everywhere. Pretty much every material has bacteria. So under the ideal conditions, which are plentiful food, moisture, and favorable temperature, these bacteria can grow quite rapidly. So basically, bacteria do reproduce and they do divide just like lots of other organisms. So bacteria can reproduce every 20 minutes under the right conditions. So basically, if we looked at the table below, one bacteria after 20 minutes will become two bacteria. The two will become four, the four will become eight. So we call the bacteria reproduction binary fission. Okay. So basically, binary fission is what we're going to be talking about in the next few slides. It is the way that they reproduce. So first of all, we have a DNA. And it's being replicated inside of a mature bacterium. Second, the cell elongates and the DNA is pulled towards the ends or the poles of the bacteria, as we can see. Fourth, the cell wall and membrane grow together, okay, as we can see. So basically, we now have two cells, don't we? And then last of all, the new cell wall fully develops, resulting in the formation of two daughter cells. So we can see here that after this is produced, one of those cells will actually divide again and then produce more cells. Now, I think this one here should have been before, but that's okay. So basically, that is the process of binary fission and all of those stages itself. Now, basically, bacteria, when we look at their reproduction, we can actually look at how fast they reproduce over a period of time. So if we can see here, we've got the log number of bacteria on the y-axis, and then we've got time on the x-axis. We can see that we've got a flat part of the curve, then a steep increase, flat again, and then a decrease. So basically, these different curves represent different things, and we'll look at what those mean. Now, basically, each part of the curve has a phase. So we call this first part here the lag phase. This is then the exponential phase. This is the stationary phase, and this is the death phase. In the beginning, the lag phase, there is a small increase to the cell number as bacteria, they get used to the new conditions that they're in. Okay, there's lots of food and the ideal conditions are met, there's going to be lots of production, and that is the exponential phase. We call it exponential growth because 1 becomes 2, 2 becomes 4, 4 becomes 8, etc., etc. Okay, it grows exponentially. After that, we have the stationary phase. This is when growth stops. The number of cells produced equals the number of cells dying. Okay, they die for a few reasons. Some of the time it's because there's a lack of nutrients and waste buildup. So if the waste buildup and nutrients decrease, there's going to be a decrease in the bacteria reproducing. Okay, so this is known as the death phase. Okay, so again, this is another picture to show it. 1, 2, 3, and 4, the 4 different stages. Okay, now what we'll do is we'll look at the conditions for growth. Now, we know that temperature itself can affect the growth rate of bacteria. Okay, every organism has adaptations for different environments. Okay, we know that already. So basically, if we look at this bacteria here, okay, this has a specific temperature in which it works at. Okay, it may be around 30, sorry, in between 10 and 30 degrees Celsius. It's actually quite good for the organism as the growth is increasing quite rapidly. Now, different organisms will have different environments and different factors, okay, but this bacteria has that, and that's quite nice, that curve. Okay, so basically, if we were to look at the similar setup, okay, we can grow bacteria on these agar plates. Now, agar plates are basically nutrients. There's nutrients inside of this jelly, this agar, and it's used to grow bacteria. We can see here that at 22 degrees Celsius, there is not much growth at all. 30 degrees, a substantial amount of growth. And then 37 degrees, the optimum, is actually a lot of growth. It means that the bacteria itself favors this 37 degrees Celsius. What is 37 degrees Celsius? It's our human body temperature. So this organism usually lives in or on our skin. Okay, and that is the end of that video. Here is what we'll focus on in the next video.